Welcome. My name is Doug Sesserman. I'm privileged to serve as the Chief Executive Officer of American Associates Ben-Gurion University of the Negev, and welcome to Hashtag Webinar Wednesday, although today we have a special edition, so it's Hashtag Webinar Tuesdays, on the race to develop a COVID-19 vaccine featuring Ben-Gurion University alumni, Dr. Tal Zaks. Before we start the webinar, I just want to thank some people: Melody Mokhtarian, our Director of Events and Experiences, Eve Rubenstein, who was so helpful in getting this set up, and also Rachel Heisler, the CEO of the Friends of Soroka, who also helped to promote this webinar. We have more than 400 participants registered, and we're now live on Facebook and believe our audience is growing to be over 1,000. For those of you who would like to share this webinar with your friends on Facebook, just post as if you were making a message and click on Watch Party, and you can share a watch party with all your friends. So thank you so much. Obviously, this is an important topic, and at Ben-Gurion University, we have two medical schools, one taught in Hebrew, the Goldman Medical School, and one taught in English, the Medical School for International Health. We refer to our doctors sometimes as the Tikkun Olam doctors, the doctors who pursue the repairing of the world. And Tal, there may be no doctor that deserves that title more than you right now at this very time. So let's get started. Dr. Tal Zaks received his medical doctorate degree as MD from the Joyce and Irving Goldman Medical School at Ben-Gurion University, and also received his PhD in immunology from BGU. As Moderna's Chief Medical Officer, Tal oversees clinical development and regulatory affairs. Moderna, as many of us have come to know, is an American biotech company located in Cambridge, Massachusetts, focused on drug discovery, drug delivery, and vaccine technology. Prior to joining Moderna, Dr. Zaks was Senior Vice President and Head of Global Oncology at Sanofi, where he was responsible for all aspects of oncology drug discovery, development, and commercialization. Dr. Zaks began his career in the private sector at GlaxoSmithKline in the genetics research group, where he built the oncology translational medicine team. He is also an adjunct associate professor of medicine at the University of Pennsylvania and has served as a volunteer physician at the Philadelphia Veterans Administrative Medical Center, treating patients with genitourinary cancers. Dr. Zaks conducted his postdoctoral research in the US at the National Institutes of Health. He completed his clinical training in internal medicine at Temple University Hospital and followed that with his fellowship in medical oncology at the University of Pennsylvania. Quite a background that built to where you find yourself today, Tal. Let's start with just the vaccine candidate. Moderna is really way ahead of the pack. There are some 165 vaccines that are in development, 30 in human trials. Moderna's vaccine candidate is now in what's called phase 3 trial. Why don't you explain a little bit about what you've learned in the prior phases and also how the vaccine actually works?
Hi, Doug. Thanks for having me. It's a real pleasure and an honor to be here, and I'm really privileged in that I found myself at the tip of the spear. Of course, I represent the work of hundreds behind me in the company. This vaccine, the unique thing about the platform here is that we actually have a way to teach the body how to make the viral proteins on its own without being infected. So it starts from the digital information of the virus, which is what enabled us to move so quickly. We were able to start the first clinical trials within a couple of months of having the sequence. We've demonstrated and shown that this vaccine seems to work in terms of eliciting the right kind of immune response. You do that by measuring the neutralizing antibodies in the blood, and we clearly get the right kind of antibodies. We do that reproducibly in everybody, and that's led us and our partners at the NIH to start the large randomized phase 3. This is the first phase 3 in the US for any candidate vaccine. We believe I'm optimistic that this will indeed prevent disease. In fact, we're We're in negotiations with many countries around the world to make sure that we can scale up manufacturing so that once we know it works, it actually is as expeditiously as possible available to those populations. I'm sort of personally happy and grateful that the state of Israel has made a deal with our company to secure some access for the Israeli population. There are several platforms out there. I believe that we will have more than one vaccine at the end of the day, but time is of the essence here. And while I wish everybody success in this mission, I think what's critical is to do this as fast as humanly and safely possible to be able to do it.
Now, my understanding of your platform and that what makes it unique is that you insert a synthetic mRNA into living cells that actually help teach or carry a message to reprogram cells to develop an immune response. Can you just talk a little bit in plain language for people like me, what that means and how it works?
Yeah, so I can talk for days about it, but in a nutshell, the central dogma of biology, and it's called the central dogma of biology, says that the way you get to protein, which is what determines what a cell does, you start with DNA, which is our genes, and you go through this temporary message that tells every cell what to make a protein. Because if you think about the cells in our body, every cell makes a different protein, yet every cell practically has the same genes. And so how do my skin cells look different than my muscle cells? Well, they make different proteins over time. And the instruction sets of which genes every cell needs to make is encoded in that transient messenger, which is called messenger RNA. So the company was founded on the notion that if you could turn that into a medicine, a therapeutic or a vaccine, you could actually, instead of having to make recombinant protein using the typical recombinant technology in artificial cells, you could just give the human body and have the human body as the factory where the medicines are made. And it's very useful for vaccines because in essence, in a way, that's what viruses do. They come in, they overtake our protein making machineries. And by the way, it's not a surprise that the most common type of virus is an RNA virus. So we've taken a chapter from evolution here, but by doing it in a very selective focused way, we're teaching the immune system only to recognize that part of the virus that it needs to recognize without having to be infected. So it's not an inactivated vaccine. It's not, it's really just demonstrating to the body, this is the protein you need to recognize to be immune. And in the case of COVID-19, it's that spike protein, which is what the virus uses to attach to cells that you want our immune system to be educated against. So we take the sequence for that spike protein, we put it in our own messenger RNA, we inject it into people's arm, just like a vaccine. It ends up going into the immune system. The immune system is now making that protein, and it's sitting there looking and saying, I've never seen this protein before. Okay, I need to go mount a response. That looks foreign. And what happens then is that if you've been vaccinated once or with a booster shot, you're then walking around with high levels of antibodies against this virus. You've never seen the virus, but your immune system has been educated based on our technology to recognize it. And so the idea is now if somebody sneezes on you and there's a whole boatload of SARS-CoV-2 there, your immune system, wait a second, I've seen that before, and can react quickly and prevent you from getting sick.
So then the human body isn't naturally immune to COVID. That's why people get sick. But if you have COVID and get over it, then you develop the antibodies on your own. And that's the concept that gets to herd immunity. And what this vaccine does is actually teach the body, for somebody who's never been sick, to develop those same antibodies.
That's exactly right. And we now have emerging proof based on several lines of reasoning that those antibodies are what's important. We know that we can transfer those antibodies and prevent illness in a whole range of animal models, anyway from mice to non-human primates. So it's the antibodies that really matter, and they've been proven to both prevent people from getting, or animals in this case, from getting infected and sick, as well as, if you're already sick, ameliorate the disease. And so there's a strong scientific basis for the expectation that these neutralizing antibodies will indeed prevent us from getting COVID-19.
And the testing you did are preclinical process with animals, et cetera. Could you just speak to that a little bit more?
Yes, so in parallel with taking this into the clinic, we've accelerated the research into all the relevant preclinical models to make sure that we are indeed getting the right immune response. We've now published in Nature the mouse data. That's a couple of weeks ago, and in fact, we got our monkey data into the New England Journal of Medicine, and that should tell you something, because that doesn't happen frequently, right? And what those models have demonstrated, that indeed we can protect those animals from getting sick. We protect them from actually having viral replication in their lungs and even in their nose. So this may even help in preventing transmission. And the level of antibodies that you see in these animals is exactly the level of animals that we see in the human volunteers that we vaccinated so far.
Just incredibly fascinating. For participants who've joined us, and thank you so many of you who have joined us, we're over 250. We had 400 pre-registered. We're on Facebook Live. We're probably in front of over 1,000 people. I can see already that we have 8 questions. For those of you who'd like to ask questions, you just click on the Q&A button. I'll be able to read your questions. And toward the end of the program, this program is called for 30 minutes in total. We have a hard stop at 2 p.m. Eastern. We'll use the last few minutes to get to your questions. So Tal, let's talk more about the vaccine, but let's go back a little bit. You're from Haifa. You're a nice Jewish boy from Israel. You were a medic.
End up doing this.
You're a medic, is my understanding, in the IDF.
Correct.
And then you choose to go to Ben-Gurion University for your medical school. Tell us a little bit about that decision. Why did you choose BGU for your medical school?
Yeah, I'm one of these guys who in high school was always into math and physics, and I went to medicine quite purposefully. My late father was, the head of the Israeli Society of Medicine, the head of the Department of Mathematics, and he was the head of the Department of Math in Haifa University. So I come from a family of mathematicians, not physicians. But I fell in love with the notion of science as it applies to medicine. It was always, for me, not theoretical like in math, but it was actually the application of science that interested me. I had applied all 4 medical schools at the time. Actually, I got into all the other 3. This one, Beersheva, I was put on the waiting list, but I was going to go there with my girlfriend, so of course I waited. There was a lady, Tarek, who was a wonderful woman. Her husband was the head of the one of the surgery departments, and they interviewed me, and I remember in one of the interviews they asked me, well, why are you coming here? We're about the patient and you seem to be much more science oriented. Are you sure this is a right fit? And frankly,
in fact, I want to come back to that, because we're known as creating the best doctors in Israel because we have a focus on the patient, the humanistic approach. And here you are, this science guy going to BGU Medical School. Go ahead.
And that was purposefully in a way because I wanted to break the mold, and I wanted to make sure that what I was doing was always, I had that aspiration for myself to understand the patient side of it, to understand the humanistic side, the side that actually for me growing up didn't necessarily come intuitively. And so purposefully for me, that was a great boon. Now, the fact of the matter is it kept me in this duality because 3 years in, I kind of look around and I said, I'm not getting enough of the science. So I took a year off. And I went to work in the lab of the late Shraga Segal. He later became dean. And I basically said, look, I want to just spend a year focusing on some scientific depth. Then I'll go back to the clinical care. And mind you, by now I was actually working 2 nights a week in the pediatric intensive care unit as a nurse. And so Soroka gave me this wonderful, wonderful mixture of anywhere from deep science at the bench to truly deep clinical experiences, nights as a nurse, physician assistant, as I was growing up to be a medic. And so when I came back into the medical school, if you will, and at the time, there wasn't such thing as an MD PhD program. Every couple of years, one Meshuggah took a year off and went to do something different. But it really allowed me this ping pong between science and medicine. And so as I was, I took another year off then before my internship, and I ended up going between the two. And I was fortunate, I think lucky, that the depth of science that Ben-Gurion had put in front of me was something called tumor immunology, which back then was sort of considered the backwater of immunology. And here we are today. It's the only curative intent therapy that we actually have for patients with cancer. And so it kind of set me on this journey of looking at the patient and looking at the science. So I came to the States to do science. I went to the National Cancer Institute, but then I backtracked into medicine in a very clinically intense area, inner city hospital in Philly. And I ended up coming out of the University of Pennsylvania, kind of doing both. And for a number of years, I was also volunteering just to keep the patient side of things going. And I think what ended up happening for me at that point is, those of you who've watched the MD PhD programs, those are long programs. And I'd spent many, many years now ping-ponging in academia and research, and it was finally time to do something in the world. And at that point, I was looking at drugs, and as an oncologist, I was always dissatisfied with the drugs that we have. But I looked at the drug labels, and if you ever open up a drug box, in the carton, there's a label, and it doesn't say this was made in the university, and it doesn't say this was made by the National Institutes of Health. They're all made in drug companies. And so my wife thought she married this good guy who was suddenly going over to the dark. Well, I mean, what are you doing going to pharma after all of that? But I felt that that's where actually the impact is greater. And the other thing that I appreciated about the move, which again, I think harkens back to a lot of things I experienced in Ben-Gurion, was the sense of a team-based effort is something that is not intuitive to many physicians. I think I was fortunate to start as a nurse and sort of experience it in depth. But actually, the pharmaceutical industry is not about writing papers. It's about getting drugs out to patients. And at the end of the day, it is a nexus of science, medicine, and frankly, business and politics too, to a certain degree. But for me, as somebody who found themselves, by virtue of the experience in Ben-Gurion, being sort of passionate about translating science into medicine, it's not science for science sake, and it's not just seeing, it's that combination. It's why I found such a productive venue within the biotech sphere and within pharma in general here.
That's so great. So Tal, I know if you could, we're blowing up on questions. We're not possibly going to be able to get to all of them already, but I know we have members of the Goldman family on listening, and we're very proud of our medical school. If there was one message you wanted to send to supporters of BGU or to the Goldman family about your experience, what would that be?
I think that your support has been incredibly valuable to the community that I experienced while I was there. I'm one of the fortunate ones. I remember the Evan Pinot when we actually had the shovel and trowel when we put down the cornerstone of that beautiful building because we were still having lessons in the pathology building before then. The ability to have and attract such a mixture of people, I mean, it's just been phenomenal. I remember the connection to the US. I mean, I think it's the faculty's connection to the US is what started me on the road to science and ultimately being able to be successful here. But it's not just that, it's the humanistic aspect that is so embedded in the faculty. We talk a lot about ethics these days, and when it comes back to the vaccine and who's gonna get it, I was taught ethics. That gap year, I did something unusual, which is I went and took an extra class in ethics. And the reason was I stumbled on a wonderful, one of the greatest teachers I've ever had, Professor Velvel Green. This guy was a microbiologist. He's one of the inventors of the laminar flow hood because he was responsible back in the day for NASA's program of infectious agents potentially coming back from the moon. But he was a religious Jew who always cared about ethics. And so when he retired from NASA, he came to the Ben-Gurion University to teach ethics. And he was my ethics professor during that year. And it's that combination. Even in that story, you can see somebody going from the deepest, highest levels of science back into the roots of what it means for actual patient care. That I credit the ability of that faculty and its supporters to make sure that they have the resources to do that. And so for that, I personally am eternally grateful.
That's great. Let's go back to COVID. So we're getting tons of questions, as I mentioned. One is about Russia. Russia's claiming that they're already vaccinating their population. Can you tell us a little bit about what you know about Russia's doing?
Very little. So look, regimes that don't operate to the same transparency standards that I think Western science has are able to do things, and they may be fine, they may not. I have no way of knowing. China is also, I think, has been in the press a couple of months ago. They've already started to vaccinate their military. There are, as I said, I hope there are more than just our vaccine approach that will work, but it's very hard to tell if you don't subject yourself to transparent, peer-reviewed, scientific rigor.
Thank you. So we're getting a lot of questions. Andrea Gorin, who's our treasurer, he said that there's a survey that reported that close to 50% of the American population claims they won't even take the vaccine. As somebody that is developing this, how do you balance efficacy in terms of the therapeutic effects with safety, and what are we learning in this trial that you're in that would ensure that the population that's healthy when inoculated would actually be safe?
Yeah, so it's a good question. There's a lot of people out there who have deeply held beliefs against vaccines for a whole bunch of reasons. Some of them reason will convince, others not. Our job is to run a trial and demonstrate the safety and efficacy, which is what we're doing to the highest possible standard. The trial that we're running is actually being monitored by an independent data safety monitoring board, a group of experts. They're not beholden to me. They're appointed by the National Institutes of Health. We ultimately report to the FDA. So it's not going to be my decision that this is safe and effective. That's actually in the hands of the FDA and independent experts. I think that's what the public rightly expects. That's sort of conservative routine practice, and that's what we're following here. So there's no surprises in that regard. You know, one of the things I personally learned is that logic doesn't necessarily persuade everybody, and I think there are other elements to earn people's trust that have to do with transparency, that have to do with communications. As somebody who translates science into medicine, I wasn't as necessarily as prepared to translate medicine into politics, which I think is sort of the question you're asking. On the merits of it, you know, there are those of us who will want to get the vaccine as soon as we can because we understand the benefit and we have an objective view on the risk. And there are those who will never want it for whatever reason, no matter what you say. And then there's the, you know, the 40% in the middle probably. The more people hesitate, you know, there's not going to be supply availability for everybody from day one. So actually from a population benefit standpoint, I'm fine with that in the sense that, you know, it's going to be, supply is going to outstrip demand for quite some time. And the longer that people go without infection, without getting vaccinated, the longer they are at risk of infection. Everything I have learned so far suggests that the benefit risk of a vaccine are going to be pretty black and white here in terms of your likelihood for benefit, given how significant the disease is. People, if they want to continue to maintain themselves at risk, you know, I don't think anybody's talking about a compulsory vaccination program. Everybody will have the data and they can make their own informed choice. My job is to make sure I generate the data and I'm as transparent as I can possibly be about that data once we have it.
So you're in the early stages of this phase 3 trial with up to 30,000 participants. Is that my understanding? Half will be receiving a placebo and half will actually get the vaccine candidate. How long does it take until you have statistically reliable results that might lead to an approval?
That's hard to predict because it's a function of how quickly do cases accumulate on the trial participants. And that depends on, you know, how big is the trial. So we went with a fairly large trial together with the US. The US is funding the trial. We're doing it with the National Institutes of Health. So it's a fairly large sample size. But again, if everybody on the trial is like me, you know, you don't go out, you don't expose yourself and you don't have to because you can work remote, then I expect event rates are going to be very low. We'll be waiting a very long time. So we're trying to gear the trial towards people at higher risk of infection by virtue of occupation and livelihood, and also at higher risk of disease should they get infected. So the elderly, people with comorbid conditions. And with that, we're also going to places in the US preferentially where we're seeing high transmission rates. So when the dust settles, I expect that by the end of the year, November, December timeframe, we should have enough cases maybe for a first interim look. And then depending on how effective the vaccine is how quickly we'll be able to declare sort of that we've crossed the statistical boundary and can actually say that this thing works.
So I just heard you say that maybe by the end of the year, you would have information on the trial results, meaning that
Correct.
vaccines could start to happen maybe in the first quarter of 2021?
I think that's our expectation, yes.
Okay, great. Amazing. So we're getting a lot of questions on side effects. What have you learned from your trials in terms of negative side effects for people who are healthy and then they get vaccinated?
So when we talk about safety of vaccine, I want to make sure people understand two different concepts. There's safety, which is side effects that are worrisome, long-term, unpredictable, and usually rare. And then there's what we call reactogenicity, which is what do you feel the day you get the vaccine and how bad is it, right? Because is it worth the pain? And the reality is that this is a prime boost, so you get first a dose and then another one 4 weeks later. After the prime, you don't really feel much, maybe a little bit of pain at the injection site. You know, a small proportion of patients have some flu-like symptoms and they go away that evening. After the boost, you can feel a little bit sicker because now your immune system has already seen this one, so it's getting woken up again. And that waking up is manifest as what you have when you have a flu-like illness. So you can get some muscle aches, fatigue, a headache, maybe a low-grade fever, and by and large, it's gone that evening or the next day, and you're fine. So yeah, you can feel something, but that is what we call solicited adverse events. They're all expected. In fact, they're collected because we ask people explicitly, did you experience headache, fatigue, da, 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 da. You go down a list. That list is what the list the FDA put out for anybody who develops a vaccine. So it's pretty standard practice. And I think the important part is it's all transient, it's all self-resolved, and we haven't seen anything unexpected or serious to date. So my
if you have underlying conditions like diabetes, are you more prone to a negative side effect? Or?
There's no reason to expect that scientifically, and I hope that with the data of the trial, we will prove that in terms of collecting that safety data on a very large data set.
Wow. Was there this is a conventional trial, was there any discussion about what they call a human challenge trial?
Yeah, there's been a lot of discussion. There's been a lot of discussion about that. I don't think you can't discern from a human challenge study what is going to be the side effects or the level of immune response in a large population. And remember, you can only challenge people with a low dose of vaccine because you can't, of the virus, because you can't make them really sick. So in a way, it could make you more confident that it should work, but it doesn't take the place of a pivotal trial. And that's why, given we've been able to move so fast, by the time the challenge studies are up and running, they'll probably be moot for what we need with our vaccine.
I've got one final question from Gary DeBode, who's our incoming board president. He's asking, are we learning with this vaccine? Are we learning good information that will help us with the virus if it morphs, or there will be another something down the road, or is your mRNA specific to COVID-19?
So our mRNA is specific to the current COVID-19. The mutants that have emerged so far and been described are not immune escape mutants in the sense that the vaccine should still protect from them, and we check that regularly based on what people are describing in the literature. That said, I do think we're learning two very important things that will serve us well if this virus will mutate or we will have a SARS-CoV-3. The first thing I expect we will learn is that this platform can indeed prevent disease. And so next time you have SARS-CoV-3, you don't have to start from scratch, phase 1, phase 2, phase 3. You basically go into production with a new sequence, just like we do for flu every year, right? The WHO says, here's this year's flu construct, and every vaccine manufacturer makes a new flu, and you don't expect them to repeat phase 3 trials. And I think the second advantage we will have is that we will have stood up a manufacturing footprint that allows us to make hundreds of millions of doses relatively quickly. And so should society encounter the next one, at least this platform should be well positioned to prevent that again. And, you know, we have an internal program in the company that we kind of affectionately dubbed the Never Again program, so that we make sure that all the learnings from this one with our platform and with the science can be applied rapidly to stop the next pandemic in its tracks and not have 2020 ever be repeated.
Thank you very much. That half an hour flew by. It's 1:58, and I know you've got a hard stop at 2. We appreciate so greatly you taking 30 minutes of your time to devote to our audience. We're very proud of you as an alum at BGU, and are hoping and praying actually that your good work will heal the world. Do you have any final closing comments, and then we'll wrap it up?
Just a deep, heartfelt thank you to the work you do and the work that you and the donors have done over the years. I, for one, have personally benefited. Hopefully, I'm giving back just a fraction of that, and I thank you for all the good work that you're doing.
Great. So for those, I know we have 58 unanswered questions. We'll do our best to get the questions to some of Tal's colleagues at Moderna, and we'll work to get answers to the degree to which we can. We really appreciate all the participants joining us. I just want to also remind people we will be back on our #webinar Wednesday schedule, Unlocking the Secrets of Plant Life, on August the 19th. And David Ben-Gurion, the founder and first Prime Minister of Israel, and the guy whose name is on the door of our university that we love and cherish, once said, "Should Israel be afraid of dreams that can transform the natural order by science, imagination, and pioneering? Of course not, for that is our vocation. the vocation of the State of Israel, and the vocation of Dr. Tal Zaks and his great work at Moderna. Thank you so much for joining us.
Thank you, Doug. I'm honored.